Imagine trying to listen to a whisper in a crowded room. That is often what it feels like when researchers are trying to capture the tiny electrical signals from your brain. These delicate brain signals, known as electroencephalography or EEG, offer an incredible window into our minds. The main sources of noise, or unwanted interference, in EEG recordings broadly fall into two categories: physiological noise, which comes from the body itself, and external noise, which originates from the surrounding environment. Understanding these helps us capture the true brain activity more effectively. Let us dive deeper into why these various interferences occur. Physiological noise is perhaps the most intrinsic challenge because the brain is not operating in a vacuum. Your muscles, especially those in the face, jaw, and neck, generate electrical signals for stronger than brainwaves. This is called electromyography or EMG. Similarly, movements and blinks create large electrical potentials, known as electroencephalography or EEG, that spread across the scalp electrodes. Even your heart's electrical activity, detected as electrocardiography or ECG, can be picked up by EEG electrodes, particularly those closer to the chest. Sweating or changes in skin conductance can also alter the electrical properties of the skin, adding another layer of biological interference. Then we have external noise. The most common culprit here is power line interference, often manifesting as a persistent hum at either 50 Hz or 60 Hz, depending on your regional electrical grid. This electromagnetic interference is everywhere. Other environmental factors include nearby electronic devices, fluorescent lights, and even static electricity. Poor electrode contact with the scalp, due to insufficient gel or improper placement, can also create high impedance noise, causing significant signal disruptions. Even slight movements of the EEG cables or the participant can introduce motion artifacts, which are often large, transient spikes in the data. To counteract these challenges, researchers employ several clever strategies. Beyond careful participant preparation and a quiet, shielded recording environment, advanced signal processing techniques are vital. For instance, digital filters can effectively remove specific frequency bands, such as the 50 or 60 Hz power line noise. Techniques like independent component analysis, or ICA, can mathematically separate out distinct sources of activity, allowing researchers to isolate and remove common artifacts like eye blinks or muscle movements while preserving the underlying brain signals. This meticulous approach is what boosts the signal-to-noise ratio, making the faint whispers of the brain more audible. Mastering the identification and mitigation of these varied noise sources is a cornerstone of quality EEG research. By understanding where these interferences come from and how they affect the data, scientists can produce cleaner, more reliable recordings. This dedication to signal integrity is paramount, ensuring that the insights we gain into brain function are accurate, robust, and truly reflective of the incredible complexity within our neural networks.